Peter, that was quite incredible. Um, and fortunately, we're not stopping. We're going to go right ahead with three distinguished panelists, and I'm going to join them as what could be called a regulatory surrogate, maybe better said. At any rate, I will try to hold up one part of this set of perspectives. And Peter, we turn again to you to moderate our session and introduce our panelists.
Well, Zan, you and your colleagues have really assembled a wonderful multidisciplinary group. Now, people often started out in the laboratory or in a more basic arena, but have differentiated in different ways, bringing us a really broad spectrum of experience and expertise for our discussion here, centered around the theme of healthy aging and longevity. So I'm going to ask Kristen Fortney to introduce herself. She is the CEO and co-founder of BioAge. Maybe you can tell us a little bit about BioAge and where you're going with that.
Thanks, Peter and Zan. Great to be here today. Just by brief way of introduction, I grew up as an aging scientist, background in aging biology before starting BioAge, where I'm the CEO and co-founder, and where we now have a program going into phase 2 and another program going into phase 1 later this year. And really the founding idea of the company, it's very much in line with what we're all discussing today, that by targeting basic mechanisms of aging, we can treat many diseases of aging and ultimately may be able to delay these diseases and extend the period of healthy lifespan. And our particular focus at BioAge is on studying what is different in populations of humans who already live healthier longer as a way to help de-risk targets as we translate them from animal models into the clinic.
Great, the extremes. And then we have, fortunately, a representative from Europe, Thomas Pieber, who's from the Medical University of Graz in Austria. And he is steeped in endocrinology and diabetes, but has also developed entrepreneurial interests and is, I think, on the threshold of some exciting consortia, which will move things forward. So, Thomas, can you please say a few words about your background?
Yeah, thank you, Peter. So I started, as you mentioned, in endocrinology. They're focusing on diabetes, type 1, type 2, obesity, and then moving into the field of metabolism. And my current work focuses on discovering or validating biomarkers for clinical studies, because I think, and I've mentioned it before, we have a huge gap between what we know from model organisms like flies or other model organisms all the way up to us humans. And we only have very few clinical trials that really tell us what to do in the field of healthy aging. So I think a major focus of our research in the future will be identifying and validating biomarkers that are a good surrogate for clinical outcomes.
Great. Graz is such a wonderful environment. And my former trainee, Andreas Zierlich, is now head of your cardiovascular center. So it's really a very rich environment, platform for your global interests. And we were also joined by Mehmood Khan, who's the Chief Executive Officer of Evolution. We already heard about him from Victor Zhao. Maybe, Mehmood, you can say a few words about your background and what your ambitions are.
I guess we're endocrine heavy. I started my career as an academic endocrinologist at the Mayo Clinic and evolved into both industry and then, of course, most recently into the nonprofit arena. Evolution's mission vision is very simple, but challenge, which is we want to expand healthy lifespan for the benefit of all. And we actually had that in our vision statement 4 years ago, which is very relevant given the discussion we just had from Victor about accessibility and equity. We have 4 major areas. We fund research predominantly in academic centers, currently funding about 200 scientists around the world. and continues to grow literally every month. We invest in startup companies, just like Kristen's, and we invest in companies from startup all the way through to pre-IPO. We are in the process of building a translational research center as a human center, and we are in the process of designing an intramural research center as well. And all of that sits under a nonprofit umbrella. So 100% of the profits from our venture fund also go back into the foundation. That really changes our approach and where we invest, the time horizon we invest in, and the types of projects we invest in. Our motto in operations is very simple. Wherever we can, we partner. We don't want to build anything that already exists, including even managing our research funding. We use partners to review our proposals. We use partners to manage the grants wherever possible, and in fact, communicate for us. So very much part of our operating approach. So that's a quick overview.
Great. And we also have in the background of the panel, two people who've been very involved with the Kitalis Institute initiatives. Larry Steinman from Stanford, who's an immunologist, and you've heard about his neuroscience program, which is going to be coming online. Maybe, Larry, if you're available, maybe you can just give us a word about what the neuroscience initiative will be that's coming up. Or maybe Zahn can remind us about that. And also Jay Schuyler, who's been very involved with the cardiometabolic area in the past. I see Jay coming on. So folks, we just had a really wide-ranging discussion with two national leaders, but here we have the academic and the entrepreneurial sectors represented, and spurred by the conversation we were having with Victor and Monica, I wonder who has some additional thoughts about how we can promote healthy longevity, working as a group and trying to break down the barriers between disciplines. Maybe, Kristen, you could start with some of your thoughts, because you're
Sure. I mean, there's certainly a lot of different bottlenecks today that we're all working on to some extent or another. There's the basic research at the very top of the funnel, right? Where there's still so little money spent on aging biology, where I feel we still have much to learn in terms of which mechanisms matter in animals and especially in humans. The part that I focus on the most at BioAge and where I think there can be a lot of innovation is really this translational gap, right? What's special about now in the last couple of years is that there's finally, I would say, a clinical pipeline for drugs that are targeting aging mechanisms. Maybe there's like a dozen or so, targets that I think that are important to longevity, that are in, real clinical trials of appropriate size, appropriate power to have definitive answers. And that's new for the field, right? Like that was not the case a decade ago. So that's wonderful. And that's why we're sort of on this upslope right now. It's still very much a drop in the bucket, right? If you look at the pipeline of any one large pharma, like a Novartis, they've got over 100 mechanisms in the clinic. So a dozen or 2 dozen is still very small, especially given the complexity of aging. So I think it's a great time. There's organizations like Evolution, right? But we really need incentives to have more people fund and test in humans these mechanisms, right? To really test these hypotheses. And that's going to be driven by the right incentives. You have to be paid at the end for these drugs that might delay or prevent disease. That's going to be driven by methods of evaluating whether they're working, like biomarkers, as Thomas mentioned, right? So that it's all linked.
So
maybe I can jump in. I think Kristen started off with a great point. Start with the science, and if I could build on that. And Victor alluded to it, human capital. How do we attract the brightest and best minds, in particular those who are early in their career, to actually take this on as part of their career? And if we look at postdoctoral funding around the world, that's been going down. And it's such a critical bridge in somebody's career. If we look at the availability of funding, even within the NIH's budget versus the $44 or $45 billion budget of the NIH, it's a tiny fraction. All of us realize the disease burden, but look at the actual funding proportionality. Then if we look at how do we get drugs to market? Well, to get them through a regulatory trial, you have to have a regulatory path. We just touch the surface of this. While I agree with Kristen, there are several drugs in clinic. They're not for the aging indication. They're all for diseases where might address the mechanism, and we hope that they will be used off-label at some point. That, to me, is not a way to attract big pharma with all of this risk. So where's the regulator in this conversation? How do we look at this? And this is a global audience. The US FDA doesn't have a track record of being very innovative as a regulator. Metformin didn't come to the US for a decade after it came to Europe. And I can go through a list of drugs that were European or UK before they were in the US. So how do we engage a global mindset? Then ultimately, how do you design a trial without an endpoint?
Right.
And so there's several bottlenecks, Peter, I think we can get into, which are going to be important in this discussion. I think Thomas's work around biomarkers is a critical piece of that.
So here's the challenge, is that studying aging is very difficult because it takes a long time, and because the endpoints would be difficult to pin down. And that's where the biomarker field becomes really paramount. And the bar, certainly for the US FDA, to promote a biomarker to a surrogate endpoint is extremely high. So, Thomas, where do you think we stand we've heard the word bottleneck, in trying to find biomarkers that can at least guide us towards what we need to implement in endpoint studies? And maybe the group could talk about what endpoints we can possibly use that could be realistic in studying aging.
So, so I do see two major problems, and I try to separate them because we have to have different perspectives of them. So one is, if we have a target, if we have a drug, I think we have a pathway in our mind coming from drug development. We know from other diseases what we have to do more or less. And even if the bars are high to get a biomarker accepted, there is a potential way to do so. The problem that I see, and that also links to the discussion we have heard before, is that other interventions are even more complex. Let's take the example of sleep and the impact of sleep or healthy sleep on aging. This is a very intriguing story. There's good cross-sectional data that there is a strong impact, but when it comes to do a controlled study, it seems almost impossible to develop an intervention, have people following that intervention, and to develop the adequate control group, not even speaking of what kind of biomarkers are adequate in that field. So I would like to separate in the healthy aging field those areas where drugs or drug-like substances, nutritional interventions are developed. There, I think we have a kind of coming from drug development, a certain understanding how we might do it. But when it comes to complex interventions, and I don't want to make it too complicated here, but when it comes to other interventions like access to healthcare or sleep or stress management, it gets almost impossible with our current means to do adequate clinical studies. And I think that's a huge challenge, and I don't have an answer. Coming back to the bar that you need for biomarkers, I think we have to have a very clear path of how we want to validate. So we start with a cross-species validation, look into animal or model organisms. Then you try to do a predictive validation and see if your biomarker correlates with an endpoint, surrogate endpoint, or a composite endpoint. I think there's a lot of work needs to be done. We need more funding in that field. But I think that there's a path, but I'm not able, or I'm looking forward hearing the opinions of others how to assess other more complex interventions. That's an intriguing part, and it needs a lot of thinking to have a path here.
Indeed, it does. He refers back to the topic of trust and what is the basis of trust. Obviously, it is based on evidence, and evidence that will be persuasive about the benefit of a healthy longevity agent is very hard to come by. We now have evidence-based interventions like good lifestyle habits, but developing a intervention that is a molecule that will affect the biology of aging is dauntingly difficult. And one of the big issues is that we don't have clear regulatory pathways. Now, we can take stepping stone approaches like Kristen is actually taking. She's targeting, and her colleagues are targeting, in effect, sarcopenia, which is a clear condition of older people, and it's actually a adversity of the GLP-1 agonist. And so if we can identify interventions, then these may also be candidates for applications in healthy populations to prevent sarcopenia. So that's the stepwise approach. But what we need is a clear set of regulatory pathways that will allow us both to repurpose already approved drugs for health span related indications and to develop rapidly, or as rapidly as can be, new molecules for which we have far less overall characterization.
So, Kristen, do you think that we're going to have to do one-off check boxes, the various kinds of endpoints that might be considered for regulatory approval? Like we have some objective measures for frailty, but are we going to have to check off cognitive function and methylation? And how are we going to assemble the data that will convince us that a particular intervention is useful for promoting healthy aging?
Yeah, I mean, I hope not, right? Because that sounds like a very long path. As Dan was just discussing, it's not as though if you have an aging drug today, clinical development, approval, and use is completely infeasible. What most of us are doing that have such medicines are in the clinic for a particular disease, right? And in our case, we're going after obesity first. And especially in many older people that are obese, they have increased risk of many different diseases of aging, whether it's diabetes, CVD, sarcopenia, et cetera, right? So there's like a very clear regulatory hurdle. There's a very clear market at the end of it. And even if initially the first label use of the drug will be for something like obesity, you can rapidly gather a lot of real world evidence to expand that label. We've seen that happen with medicines like statins. So there is a path today. And of course, there can be a better path, right? I think there's a lot of work insofar as biomarkers are concerned. I think there's good news and there's bad news. The good news is that I really think the field has made tremendous strides in the past decades. We used to talk about things like telomere length as being good predictors of aging, and they're not, right? They're incredibly noisy. They're not very predictive. They have very low predictive power. And now, whether it's the methylome or the proteome or the metabolome, these sort of broader sets of measures that are really more, system-wide, are incredibly predictive. And that's really good news. Like, I think you can get a lot of, and you can argue about whose methylome biomarker is better, but they're honestly like quite useful right now from like a practical standpoint. I guess I do not think it's likely they're going to be useful as a surrogate endpoint in the near term. I mean, we've seen this play out with osteoporosis, with cholesterol. Like that's a very long path. I think nevertheless, they can be useful as research tools in the clinic to sort of help build evidence and conviction that your mechanism is behaving the way you think it is, that it's acting more broadly as an aging drug, as opposed to just being efficacious, on your primary endpoint for this one particular disease. So I think that we're still getting a lot of value from them, despite the fact that, a surrogate endpoint might be some ways away.
Yeah, so we're hearing that there's a very high bar for regulatory approval for a drug, but there's this huge disconnect because supplements can get on the market making thinly veiled claims for improving health outcomes and longevity without much regulatory scrutiny. And is this a problem for our field, and how can we address that? Because I think the public is all too willing, and I mentioned this earlier when I was speaking about the frayed trust with the public. and organized science and medicine and research in general. How can we address the change in belief systems so that we can get people to embrace evidence-based medicine? And how do we get regulatory pathways to come better into conformance with behavior of people? I mean, these are huge societal issues. I mean, the supplement industry is enormously lucrative, but is bolstered by scant data in terms of outcomes. And I think we've done a bad job of educating the public. So what can we do better, folks?
Well, Peter, I think it's going to be very hard to put the dietary supplements back into the bottle, so to speak. They're going to continue to be out there, and it's not likely that there will be any change in the environment. But what we can do, as you say, is educate. And the other part is making the public more aware that there is scientific progress being made in these areas for which they are taking supplements. So it gives them some frame of reference to see that scientists are actually producing data to demonstrate that a product works for a age-related condition. So I think in a nutshell, it is education, but it's also making the public confident that scientists and others are on the job, that we are taking this on, and that we're all in it together.
So, Mehmood, what steps is evolution taking, and you in the broader sense, towards educating the public about the progress that we're making and the challenges that we face in bringing the innovations to the clinic?
So I think if we take the public perspective, you sort of can start to empathize. First of all, unlike diabetes or cardiovascular disease, the field of neuroscience and aging doesn't have a professional body. It's not like you can Google and go on and say, what did the experts think? And the expert community is far from a consensus. And we have a spectrum of people that some would consider are at the extreme of sort of pixie dust, magic, all the way through, to spas, through questionable ethical practices, and we think about blood plasma transfusions, through to credible science. And I think one of the challenges we've observed in a gathering like this is this is an informed audience and an informed gathering. The challenge is not people on this panel or certainly this audience. So how do we get this anchor in science? The other thing we found, and we actually did some market insight work across the world in dozens of countries. And we held focus groups in North America, the UK, and Asia, and started to ask some of these questions. Another time this data is available from us publicly. We've been publishing Global Health Span report now, and it's very insightful. And we start there. Listen to what people actually are hearing. The second thing is, last year in the fall, we convened a gathering of experts around aging from the pharmaceutical industry, academia, regulators, biotech industry, investors, bioethicists, and we thought we'd get a couple of hundred people come together. We had to close the registration at 2,000 people. And that tells me that the demand for such a meeting and a convening was there. We didn't do anything magical. We just brought the people. And for the first time, we had a regulator, a venture capitalist, a biotechnology CEO, all on the same panel, having a conversation. And I think that type, not the only one, there should be several, where we can bring credible people, credible in the sense of anchored in the science, not just with one opinion. And I think that forum has been lacking. I just gave you the number that pretty well demonstrates. And this was 2,000 people flying from all over the world, not from one country. And I guess if we had the capacity, could have gone to 3,000. We just didn't have the space. We had planned on about 500 or 600 people. And it was a huge mistake on my part as CEO. So that's another. The third is we need to start to communicate the evidence. And the evidence is not getting out there. We do not have a definition of what is a good biomarker. We don't have a consistent approach on what does geroscience mean? What is the difference between geroscience and geriatrics? Most people who hear geroscience, they hear geriatrics. And so there's a lot of confusion. We can go into that. And I think communication is a big part of this right now. And the scientific community is not all on the same page. No, I'm not suggesting that everybody should have the same opinion, but we should be able to figure out, just like my former life as a membership of the American Diabetes Association, yours, American Heart Association. Not everybody at the HA disagrees, but there's sort of a consensus. I don't see that in this space.
Mhm. So, Thomas, we've spoken about the lack of a biomarker which has been elevated to surrogacy. What would be your top couple of candidates if you wanted to put down your money on the board there? What do you think is the most promising as a biomarker which we might be able to hang our hat on for surrogacy?
Yeah, so the approach we are suggesting and have submitted to our funding agencies is that we would like to move from disease-specific biomarkers into more general ones of aging. So I think type 2 diabetes, obesity, healthy aging is a wonderful example, and the GLP-1 story that has been mentioned actually tells us that we are looking at the same mechanism. So you could imagine that and forgive Peter if I'm starting with your field of expertise, but you could imagine that for heart failure, you start studying interventions in a very specific group that has advanced aging of the heart, early heart failure for genetic reasons or for other reasons. You study, for example, the impact of obesity or weight loss in that specific group and try to expand then to a larger population and then to the general population. That's at least together with our cardiology colleagues here in our university, one of the approaches we are trying to do. At the same time, we have to introduce multi-omics, as costly as it is. And there, I think metabolomics profiling or using a metabolomic fingerprint seems to be promising. So not analyzing all details of, I don't know, 2000 metabolites, that's impossible. But there's the concept of a metabolome profile, like a fingerprint that indicates a healthy status compared to the disease status. Of course, again, you start with the disease, and this comes now from cancer in oncology. So I think we have to start in a specialized field where we do understand the disease mechanism and then broaden it and try to validate it in a larger population. That's at least the approach that we are taking here in Europe. If that succeeds, it's difficult to say. If that succeeds when it comes to regulatory purposes, it's very difficult to say. We had discussions with EMA, the pendant to the FDA, and they are, of course, talking to each other, as we know. But it's difficult to get concrete advice in the current stage because I think we have to acknowledge there's not a clear concept yet available for those kind of interventions. So it will take us some time and substantial research to get that field moving. But surrogate biomarkers helped us, and the LDL and the statin use. We also started using statins in people with coronary artery disease. And then we're expanding into prevention, primary prevention, and now we're looking into the impact in healthy aging. GLP-1 very similar. So GLP-1 started as a diabetes medication. Learning has an impact on satiety, and now we're expanding it to a broader population. So I think if we have a good intervention, it's easier to identify biomarkers that could be translated into a larger field. If we start at healthy aging itself, that's a critical one. So the clock genes are still good candidates, methylation status, but there's again coming new data on metabolomics profiling that are quite interesting.
And that really is the big challenge, is that you can't develop a surrogate endpoint in a vacuum. you actually need an intervention that you can use to confirm that it reliably predicts clinical benefit. And we've got history here with probably the two best well-known surrogate markers, LDL cholesterol and hemoglobin A1C. There was a day when we did not have those, and that was just as I was in training as an endocrinologist. We had a lot of epidemiologic data that LDL cholesterol was important in the development of cardiovascular disease. And when I was at FDA, my first review was of the first statin, and we approved that drug on just two small studies that had LDL cholesterol as the endpoint. And yet it took decades to get to that point and an effective intervention to confirm it. Likewise, for hemoglobin A1C, it took the landmark trials, the Diabetes Control and Complications trial and the UK PDS study, to confirm that A1C actually predicted clinical benefit, though we were approving drugs on the basis of A1C before we had that confirmation. But the point is, it took a long time, and we somehow need to compress that time for validating biomarkers as primary efficacy endpoints.
You know, it's very interesting to look at the history of the hemoglobin A1C as a approvable surrogate biomarker or surrogate endpoint. I think that I credit my friend Stephen Nissen as doing the right thing for the wrong reason, because he advocated with the Food and Drug Administration in the US to require a cardiovascular safety study because of concerns about rosiglitazone safety. And there was initially a huge uproar and hue and cry from several sectors, the academic and the pharmaceutical industry, that this would kill all drug development. And if we hadn't had that guidance from the FDA, we wouldn't know about many of the cardiovascular benefits of the incretin mimetics. And we haven't mentioned here what's transformed my field is the SGLT2 inhibitors. So this is a real testament to the biomarker field and how rigorous we have to be for requiring safety studies as well as efficacy studies on biomarkers. And of course, we were vastly misled in my field by stamping out ventricular premature depolarizations, because we found when the study called CAST was done that we were actually harming people by addressing the biomarker that we had faith was going to reflect cardiovascular outcomes. So we have to err on the side of caution when we're thinking about elevating biomarkers to surrogacy as well.
And by the way, Peter, you mentioned two classes which are actually at the top of the list of candidates for not just managing chronic diseases, but actually preventing them: the SGLT2 inhibitors and the GLP-1 agonists. Jay Schuyler has been very much involved from the beginning, and he mentioned earlier on that he was even a patient early on with the first generation of the GLP-1 agonist, and he's reached a 20th year anniversary of being on that class. So we've got candidates, but we need to put them to test, and to do that, we need to decide on endpoints that would allow for a multi-morbidity indication to be granted by regulatory agencies.
But it's interesting, the three classes we've mentioned were all on the market for a therapeutic indication, and then developed the data, to Kristen's earlier point, on preventive. So they didn't come on the market as preventive, whether it was statins or SGLT, GLP-1. And so it still begs the question, is there a way? I don't know if there is there a way of accelerating to actually bring a therapeutic to market as a preventive. I can't think of one in the drug domain which came on with that as an indication for a prevention.
That's right. And I think we could hope that drugs that do show some promise for modifying the aging process that are approved and do have established safety profiles could be tested and shown to be effective for reducing the risk of multiple chronic diseases. Metformin, which I also approved at FDA at that time against a lot of opposition, is now a favored candidate for improving healthy longevity. But we need a trial to show that it works. And to do that, it will take an accepted endpoint and probably a pretty big trial and even a long one.
So, we have.
And Zan, that there's a lot of enthusiasm to fund such a trial. It's just getting all of the pieces together.
Well, that's right. But we need more shots on goal. And evolution, of course, is one big help for that. And we're going to need organizations like Evolution, along with NIH and other government agencies to support the kind of trials that are needed.
Well, there's some very vocal leaders in our field who think that the randomized clinical trial is not the only way to try to show efficacy of an agent or intervention, and that we can use big data and genetic analyses and large data sets to try to converge on clinical truth. And I wonder whether panelists think that the randomized clinical trial should remain the gold standard, or whether we can mine big data and the electronic health record and observational studies as a way to gain confidence that a particular intervention is actually efficacious.
Well,
I mean,
difficult.
go ahead, Thomas.
Sorry. So I think when we come from the field of drug development and a randomized controlled trial with proper control, being placebo or best practice or whatever you have, will remain a standard. And if you take this example of SGLT2 inhibitors again, I mean, they were developed for a very specific purpose to increase the glucosuria in type 2 diabetes, and it turned out to have other impacts when you study it. And then you can expand your trials. And of course, you can imagine doing an intervention trial with SGLT2 inhibitors in a healthy population in the near future. So I think it depends also on the magnitude of the effect you're achieving with your compound or with your intervention. That makes a big difference. So if you have only incremental improvements, it will be very, very difficult to continue with randomized controlled trials, and big data get more attractive. But I think we have to combine both worlds, so have randomized controlled trials and then expand with big data to other fields of populations could be a way out of that kind of dilemma we're in.
So other people want to speak to big data analysis as being a savior for our problems?
Not a savior, of course, right? And I don't think RCTs are going anywhere, but being in biotech, I take a very practical perspective. And can it mean spending a whole lot less money and taking on a whole lot less risk? Yes, right? Like big data, EHRs can help you come up with a very targeted initial hypothesis. So maybe your first efficacy trial doesn't have to have 2,000 people, it can have the right 200 people. And then once you see that initial signal, you can do a bigger population trial, put in more funds, invest more dollars, right? But I think that it can help save the field a tremendous amount of money by having us, create very focused hypotheses about particular agents. I mean, some of the metformin hypotheses came from observational data, right? That's a big data hypothesis, as well as, selecting patient subpopulations most likely to benefit. So you might see, a bigger initial signal and have more power in your first small trial. So it's going to help make everything more efficient, certainly, even without replacing RCTs.
Yeah, well, that's certainly an established use in my field of biomarkers, is to select populations for which a specific intervention might be beneficial, and to enrich your population in a clinical trial for response to a particular kind of intervention. So there's some, I'm scrolling through some of the chat, and there's some very provocative and interesting questions online. There are a couple of posts here in the chat by Ben Wiegand, talking about how we're going to get people to adopt healthy lifestyles rather than small molecule or biological therapeutics that are inevitably expensive and difficult to get approved in a regulatory environment. So how do we address behavioral, social, and and psychological solutions as well as our drug interventions?
Peter, I would, first of all, violently agree that we need to avail ourselves of interventions that we know now work. It's just they're hard, but we know that keeping at a normal body weight and managing other risk factors, getting good sleep, having good social interactions, all these contribute to greater survival and reduced adversity. So the problem is it's tough medicine for most folks to do what they know is a good thing to do. And I'm not the expert on how we modify the human behavior that results in lifestyle choices that are not so good for us.
Let me perhaps build on that. I think Zan's absolutely right. I think there's a huge gap between what should be done versus getting people to do it. I don't care which field it is, whether it's cardiovascular, endocrine, right? We've all, and how do you bridge that translation into the real world? We can all do it on the GCRC, or I don't know if they're still called GCRCs. I'm dating myself. We can all do it there, but do you actually get it in? That's one thing. The second is, I think our understanding of behavior is also changing. Look, we used to think that obesity had a high degree of choice. And then GLP-1 agonists came along. And so there's a clear interaction between the brain and other parameters. We don't know everything, but it has raised questions around how we make choices. And the third thing that hasn't been brought up, but it has been brought up in the chat, is that we know, for example, patients with diabetes do better when they get real-time feedback. And so what is the role of feedback loops? Whether it's on a specific variable like glucose, I don't know if you could do this in real time with hypertension. I suspect you could do to some extent. And the technology is getting better and better. And the biggest devices in the healthcare monitoring are unregulated. And consumer industry has figured out a way around it by not calling it a medical device. And I think, Zan, you have a lot of experiences. It still raises the question, how do you get them into the fold so the data is accessible, right? It's sitting right now. It's a pity. It's siloed. But it could be more in the public domain, and we might be able to do a lot more in this interaction between the drug, the behavior, the activity, and whether it's wearables, implantables, or wherever these things might go. In some that we think of, closed loops with insulin, but what about the future closed loops of other variables? And so I know I'm posing a lot of questions there, but I think that we're getting closer on the questions. And the last point I want to make is, if we're going to make these therapeutics better, then we have to get away from the averages of the population. We have to start to understand with much more specificity on which of the subpopulations are going to benefit the most. That, of course, not only brings the size of the trial down, brings the cost down, but it also eliminates a lot of the wastage. We all have seen the data of the percentage of the population that responds to a statin, right? How do you get down to that? So there's a number of these things, in particular for long-term effects, where you're not going to know if it worked until a long time has passed. Unlike your LDL, you'll know in 90 days if somebody's really responding. So I'll just stop there. I think there's some interesting areas and relevant to evolution. We're asking all those questions right now. We're asking those questions, and the other point we want to make is, You're not going to get diverse populations in the aging area just by diversity of genetics. It's going to be diversity of location, because aging in the environment you age in probably plays an important role regardless of, an Asian American like myself living in Greenwich, Connecticut is very different than my ancestral cousins living in the Punjab in India. I might be genetically the same, but I'm certainly not in the same environment. And the British studies on Indian immigrants has clearly demonstrated that they're in a very different biological path than their relatives. So how do you fund studies across borders? And how do you pool that data? The data might be available. How do we bring it together? So I think there's a lot of opportunity. Those are sorts of questions we're starting to ask at Evolution in order to not be limited by political boundaries.
Right.
Those are such great points. And I would just pick up on the example of continuous glucose monitoring. Jay Schuyler has been involved from earliest days with that technology, but it has revolutionized the care of people with type 1 diabetes, and it's increasingly being used for type 2 diabetes. But the value may ultimately be in managing weight. Having that immediate feedback, as you say, is very important for affecting good behavior. If you don't have that immediate feedback, it becomes a much tougher proposition. So it's a great example.
Yeah, well, there are more and more people using the technology without diabetes. You point out for control of weight, but athletes are using it to look at their exercise thing. And people are looking at the variation in their glucose levels to make all sorts of decisions. And an over-the-counter version of CGM, which doesn't require a prescription, will be available this summer. And I suspect there'll be I've talked to a number of people who say, well, I just want to try it and see, learn some things about myself. So I think we're going to see more and more of use of sensors that not only look at glucose, but look at other substances that are going to help us determine what's happening and getting a better profile. Whether they can be used toward clinical endpoints will be more complicated because you have to have a clinical endpoint that you're relating whatever biomarker you are trying to promote to a surrogate marker for. And that's not so easy to do. And you have to have a clinical endpoint for aging and for healthy aging is something that is more elusive.
Can still be a great intermediate though, right? Just to continue on this theme of these continuous metrics. Like you have to be able to measure what you're doing to know if you're having an effect. That's why we have so many approved drugs in dermatology. You can like really see what you're doing, right? And we were touching earlier on sarcopenia and frailty. And one of the reasons that's historically been so challenging to develop in is because what's the approvable endpoint at the end of the day? It's like a one-off 6-minute walk test or something of that flavor, where there's this, huge variability in, site to site or how you happen to feel that particular day. And even something as simple as a wearable, right? And that kind of data, I think, could make that a much more tractable indication. So this isn't healthy aging itself, but some of these indications that are, I would say, in between conventional diseases and aging itself that are going to be made more tractable by these types of devices, that I'm excited about that too.
So this conversation is giving me some optimism because taking the lesson from glucose monitoring in type 1 diabetics, providing feedback which can improve their health behaviors, maybe that is the angle to enlist the broader public in healthy behaviors, because with the dissemination of wearables, with the banality now of smartphones, maybe that's the way that we're going to close the circle and address the skepticism of the public in science if they are involved themselves in monitoring various health variables. Maybe that can incent good behavior, and maybe that is a way for us to climb out of this valley of disbelief in which we seem to live. Certainly I, in my hospital practice, am confronted not only with the all-comers that I see with my fellows when I precept clinic, but also even in my resourced and educated patients who say, well, I really want to try 6 months of diet and exercise before I take your pharmaceutical agent, and it's a conversation that is never ending with my higher echelon patients. So I think that, as I say, this conversation is giving me some optimism and showing a way forward. So, if rural farmers in India can get quotations for commodities on their cell phone, maybe they can count their steps and engage in other health behaviors that would be salutary.
And Peter, on that,
the other thing
ahead.
is they can get. Go ahead.
I was just going to say that what we're seeing is also a generational shift in consumers. I'm going to use the word consumer, not patient, willing to share their data from our generation, Kristen excluded for a minute, to a younger generation that seem to be willing to share everything that they're doing in every aspect of their life. It blows me away. We're also seeing this difference in willingness to share across in different countries. And so I think how to engage that trust level, given that generational shift, but at the same time, the previous panelists made some excellent points around building trust. The more we can do that and get for the common good, look at the businesses that have been collecting data and people have been willing to give that data up because they got feedback. I now know that I have 1% Neanderthal genes because I had my DNA looked at. Was I willing to give that up for that information? It wasn't for the Neanderthal part, but the rest of it, so I was curious. So, and I think we're going to see more of that. My point is, I'd love to see the regulated part of our society and the unregulated part come closer and figure out.
You actually touch on a very powerful tool, and that is big data, which is afforded by the technology of having smartphones and wearables. And we could imagine that randomized trials that would consist of 100,000 people in each treatment group could be done with in a case of a sarcopenia indication that Kristen might develop, would look at an accelerometer function that's built into the phone and would read out on gait speed, which is a very good surrogate for survival. So big data can be used and it can be done in a randomized way, which is really the gold standard.
The other thing that happens with smart devices is they can help people do things different. While we were sitting here, my smartphone, smart watch just went off and told me it's time to stand and move around.
Right.
But these things can help get people to do the things that they need to do to improve their health and the lifestyle component of it. I think it's crucial.
It cuts the distrusted professional out of the loop, and I think that may well be a way forward. And perhaps the younger generation overshares from some of our perspectives, but I think that really is a very productive perspective, and it grows out of the endocrine community. And wearables are really transforming a lot of arrhythmia monitoring in current environment, and the Apple Watch study was an example. It was led by Mehmood in Larry Steinman's institution at Stanford. So we're democratizing and going directly to the consumer for ways in which we might be able to establish better health behaviors. And then also monitor the biological variables that will help us to dose modify some of our interventions.
I think Thomas Sir might be monitoring the chat as well, and he could maybe relate a few questions from the audience. So this is a good opportunity
One
to.
weighty comment, I mean, a topic is, do any of the panelists consider the microbiome as a potentially targetable mediator for healthful aging? Big topic.
Yeah, well, Monica did
Yes.
mention that, and certainly there's a tremendous literature that is burgeoning on the microbiome based on the technology that allows us to look at the microbiome and dysbiosis. From my perspective as a journal editor, there's a lot of lack of concordance in the various studies. So I think we may be in the infancy, but clearly there are many more bacterial cells than human cells in our bodies, and a considerable amount of our body mass is made up of the microbiome. And the microbiome is definitely involved in altering metabolism or in elaborating metabolites, which may be influential in behavior of cells. So I think that it's a pregnant area, but I'd be really interested in comments of others, because I've seen a lot of lack of reproducibility or lack of concordance in various studies with the microbiome.
So at Evolution have received numerous applications for research with both research proposals. We've looked in total already about 200 biotech companies that have approached us for investments, some in the microbiome space. So I'll sort of summarize at least what we're seeing out there. As you mentioned, Peter, and I think as we're all aware, there's a lot of association patterns we see between different microbiome makeup and chronic diseases and different physiological states, whether it's obesity, etc. The challenge, if I sort of simplify, and this is an oversimplification, is one, demonstrating a cause and effect through the classic Koch postulates. And again, I'm dating myself here, but number one. And then the number two is, if you were to develop an intervention, how do you actually define the intervention in dose, consistency? It's one thing to have a product that has a probiotic, but we're talking about, and we'll leave Clostridium aside for a second, but we're talking about an intervention that says you take this mixture of bacteria or microorganisms, or we eliminate the following microorganisms very specifically, then your biological state will change. That's a really tough order. And we haven't yet seen, I'm not saying it's impossible, we haven't seen something that our review panels, both scientists or investment panels, have been comfortable to saying, yes, this is a clear approach. I'm not saying it's not possible, we just haven't seen it. I'd love to know if others have taken this knowledge. I thought I'd give you sort of a science proposal / investment approach overview.
Well, if I may step in here. So first of all, it's a new universe we are opening, which we were ignoring 20 years ago, more or less, because we didn't understand it and we just left it out. I think it's quite clear that we as human beings, as any vertebrates, are in close interaction with the microbiome in us and around us. I think one of the major problems is that what we consider what we are measuring currently is a fraction. It's mainly limited to stool. It's limited by the sampling technique. It's limited by the analytical approach. It's pretty intriguing. The first companies are starting developing new devices where you can sample microbiome, like in the stomach and then in the small intestine, because that is probably more relevant to as for the metabolites and the immune interaction with the immune system than the stool only, which is kind of the end product in a certain sense. So I think we just started understanding that world, and it will take a lot of standardization and analytical validation before we can go into the question, is there a cause relationship, or can we intervene with the microbiome and can we have an impact? So I think that's too early, but it's clear that any healthy aging has to include the world of the microbiome. Otherwise, we are leaving out an important part of our environment.
Can I introduce another question? George Radenberg of the Davos Alzheimer's Collaborative pointed out that he's very active in looking at ways to find vaccines for Alzheimer's, and there are vaccines for cancer. These are one example of preventives that have been approved. And Alexander Kissen also asked the question, what would an endpoint around cancer prevention look like for these metabolic drugs like statins, GLP-1s, SGLT2s? They haven't been shown to prevent age-related cancers or diabetes. So maybe people could comment on the preventive approach. We are trying to preempt chronic diseases.
Yeah, I wanted to mention vaccines or vaccinations before, because when we were discussing, we don't have a drug approved in a healthy population. Actually, vaccines are the only drug we currently have in our system that are approved in preventing a disease only and doing nothing else, which is interesting. So it's interesting to transfer that concept from infectious diseases only into chronic diseases, which might still have an infectious root. We are not really sure, or we are learning more and more how viromes and other microbiomes are influencing our genome. So that's an interesting question. I cannot answer that specific question, but I just wanted to highlight that vaccination is the one and only example we have in our healthcare system where we prevent the disease in a healthy population. And we even have methods to measure and assess the efficacy of it. And COVID just gave us the latest example how effective that can be. Yeah, with that, I will leave it to others. Maybe they are more.
I mentioned that contraception is another preventive, but obviously that's not a disease, but it is a preventive that's been approved.
Well,
I'd appreciate hearing Zen Fleming's perspective. I mean, given your experience in this, because I think it's as much a science question as a path to get this done.
San, are you frozen?
I think he froze there.
Right. Well, Mehmood, as you know, if I may be having
little internet interruption issues there on Zan's part.
Mahmoud, we can take that offline, but it's a very
issues.
Go ahead,
Can you
Sam.
hear me now?
Yes,
Yeah.
not now.
Yeah. Well, I think that we have to, we're actually rounding on the hour, and I think we really have to.
Well, I don't know. I
Sam.
apologize for my internet.
We have to thank Zan for bringing us together. I think we could go on until cocktail hour, depending on what time zone you're in. But I do think that we have to, out of respect for the audience, wrap this wide-ranging discussion up. And as I said, I think I'm ending it on a note of optimism. I think that we stand on the threshold of a time when we're really going to be harnessing some of the basic science advances, harvest the fruit of the research, and try and move it forward to promoting healthful aging. And I think that we thank all of the panelists for their wonderful contributions from taking the time to help us here. And I don't know if Dan is able to give a concluding remark. Okay, well,
So let me step in with a housekeeping.
I don't know if you're hearing me now.
We can now. Go ahead. Whenever we wait for you, it freezes. I'm sorry. Let me get the housekeeping out, and maybe Zan will be back with this connection.
Well, I'll be brief.
why don't you turn
Thanks.
off video?
Yeah.
Dan,
Yeah.
turn off your video. That can
Go
sometimes
ahead.
help with your bandwidth.
So just a
I
reminder
will do
that.
that. Go ahead, Thomas.
Okay, just a
And
reminder
heartily
that.
thank.
Absolutely. Just a housekeeping comment to finish that, again, all registrants will receive the link to the recording within a day or two, and then links to the slides, the transcript, and the chat log thereafter. I want to remind people that our Targeting Healthy Longevity 2024 Session 2 will be on June 7 on novel interventions for preempting neurodegeneration, moderated by Larry Steinman, seen here before from Stanford, as well as Harvard's Lee Rubin. And I know that they're seeking confirmation from some stellar panelists, so block that time, please. All registrants to this session 1 will receive information on the next session in Targeting Healthy Longevity 2024 by email, so please be on the lookout. And with that, we certainly thank the speakers and you, the audience, for your attendance, and wish you all a good day. Have a great weekend, wherever you are around the globe. just speakers and attendees, the virtual hall will be left open for a few minutes for those of you who are available and want to tarry. So Zan, I don't know if you want to try to come back in now, but I would say that the formal part of this call webinar is closed. Zan?